My name is Edward Catoiu, and I'm a graduate student in Dr. Paulson's lab. Welcome to a recorded lecture for BENG 212 for the winter quarter of 2020. We are recording this lecture in an effort to transition and adhere to UCSD's policies restricting in-person lectures for the upcoming spring quarter. This lecture is an introduction to structural systems biology, and I hope this presentation will help familiarize you with some of the many research applications for protein structures. First, we will look at what protein structures are and what information they contain. We will then show how protein structures can offer mechanistic insights into biological processes with angstrom-level detail. In doing so, we will look at examples of viral mechanisms, including ones relevant to the recently defined coronavirus pandemic. Furthermore, we'll examine some applications of protein structures when combined with genome-scale modeling. And finally, we will briefly discuss future prospects for the exciting field of structural biology. With no further ado, let's begin. The outline of this lecture will be as follows. First, we will go over some relevant background information about proteins and provide a brief biological review. Next, we will discuss how protein structural data is determined, namely, how do we even know what proteins look like? Then I hope to give you a brief glimpse into the level of detail with which we can understand biological mechanisms using these same structures. Next, we will take a look at the protein information that can be calculated in silico or mapped from experimental databases at the genome scale. And finally, we will look at some applications of protein structures integrated with genome scale modeling. Proteins are the final step of the central dogma, which, as we know, describes how the genome contains genes which are read and transcribed into mRNA by RNA polymerase in a process known as transcription. This mRNA is subsequently read and interpreted by tRNAs in the ribosome to form a polypeptide sequence in a process known as translation. And lastly, the polypeptide chain that's been formed folds upon itself in its finalized form as a final protein, which is now ready to begin cellular work. But what do we mean by cellular work? Well, let's say we wanted to build a house. We're going to need the right tools for the job. But let's also say that when we open our toolbox, we find things like silverware, an apron, a chef's hat, and spices. In this case, we are probably more suited for cooking a meal than building a house because we don't have the right tools. Similarly, let's say we now open our toolbox and we find all the tools that we need, except that instead of nails, we find thumbtacks. Let's even say that we could still build a house held up by thumbtacks. It is obvious that the results of our work will likely be less reliable and eventually lead to problems down the road. In a cell, proteins act like the tools in our toolbox. If a cell doesn't have the right tools, or if the tools are damaged in the slightest, this can lead to unintended results. Let's take the red blood cell's job to transport oxygen throughout the body. For this job, the red blood cells must have free hemoglobin that can carry oxygen. Additionally, the red blood cells themselves must be able to freely and rapidly flow through the circulatory system to deliver oxygen to where it is needed most. Now let's say that one part of this hemoglobin is mutated, changing a hydrophilic amino acid into a hydrophobic one. What happens? Well, in sickle cell anemia, this is precisely the case. Glutamic acid at position 6, shown here in purple, is mutated to valine, a hydrophobic amino acid. This hydrophobic amino acid lies at the protein surface, thus it is readily exposed to the aqueous environment. To avoid water, it interacts with other hemoglobin proteins, forming irregular, elongated chains of weakly bound hemoglobin. At the cellular level, these elongated hemoglobin chains distort the shape of the red blood cell and prevent normal blood flow, increasing the risk of clots and other complications. Rather than saying sickle cell anemia is caused by or associated with a specific point mutation, this is one example where protein structures can offer detailed mechanistic insight into exactly why mutated hemoglobin causes red blood cell sickling and eventually leads to the disease state. Understanding that proteins are the tools that the cell has to carry out biological functions, what else are proteins capable of? Well, they can act as reaction catalysts, transport nutrients in or out of the cell, regulate the transcription of genes or even the activity of other proteins, sense the external environment and signal this information to the cell, and they can provide structure to a cell, such as a cytoskeleton in eukaryotic cells. The extent to which proteins are able to carry out cellular work can also be modified or tuned through allosteric regulation, which is the binding of a protein or small molecule to a regulatory site on the protein separate from its active site. Their behavior can also be modified through post-translational modifications, such as phosphorylations that can turn these enzymes on or off. They can also be modified by their own ability to oligomerize into structures with multiple subunits. At what scale do proteins operate? From a top-down level, an individual is comprised of a network of organs that work together to maintain homeostasis. Within each organ are specialized cells that have specific functions vital to the functioning of the organ. Each of these cells, in turn, has its own molecular network capable of carrying out chemical reactions, turning substrates into byproducts. And lastly, the architecture of this network is determined by the genes present in each cell and whether or not these genes are expressed at any given time. Proteins, the tools with which the cell carries out its reactions, operate at the angstrom level. For reference, the length of an E. coli cell is around 20,000 angstroms, while the diameter of the average protein is on the order of 50 angstroms. Additionally, the distance between amino acid side chains in a protein is usually 7 angstroms, while the length of a single carbon-carbon bond is around 1.5 angstroms. So why should we care to study proteins? For starters, here is a list of the top grossing pharmaceutical drugs sold in 2015. If you look closely, 7 out of the top 10 pharmaceutical drugs in 2015 were proteins. Don't see it? Here's what they look like. Not only is structural biology a lucrative field, but it can also have many human health applications. If you're more motivated by academic pursuits, structural biology is really for the scientist that likes to ask why things happen the way they do. In this manner, protein structures can provide mechanistic understanding of cellular processes at the angstrom level. They can also help us understand drug pharmacological and kinetic properties. And lastly, by studying the way protein structures interact with their substrates, we can begin to understand network regulation at the system scale. Before we can work with protein structures, we're going to review the basics. We'll go over the building blocks, which are amino acids, and we're going to look at their biochemical properties. We're also going to look at how these amino acids assemble into their primary, secondary, tertiary, and quaternary structure. We'll also examine some of the patterns that are commonly found in these structures. And lastly, we'll look at the shape of proteins and determine how do we know what they actually look like. The fundamental units of proteins, or their building blocks, are called amino acids. There are 20 amino acids, and each one differs based on which side chain it contains. For example, glycine contains no side chain, making this amino acid extremely flexible. Glutamate, on the other hand, has a side chain that's very negative. The variability in the side chains of amino acids give rise to their variability in their amino acid properties, such as size, shape, hydrophobicity, charge, reactivity, and flexibility, among many others. From the central dogma, we know that as the mRNA gets translated by the ribosome, amino acids are sequentially joined to form a polypeptide chain. As the chain grows, it folds onto itself and can associate with other folded polypeptides to form the final protein structure. Protein structure is often discussed in terms of levels that reflect the structural complexity. The primary structure, or sometimes just called the primary sequence, is just as it sounds. It is the amino acid sequence, which is held together by peptide bonds. As the polypeptide sequence folds onto itself, local patterns form. In this example, an alpha helix is shown. These local patterns are held together by hydrogen bonds. The final structure, or shape, for that polypeptide sequence is referred to as the tertiary structure. This is held together by hydrophobic interactions, salt bridges, or even disulfide bonds. As these tertiary structures interact with each other, they form quaternary structures, which refers to the multi-subunit architecture of many enzyme complexes. These are held together by non-covalent bonds. As the shape of more protein structures became known, patterns emerged. Patterns found in secondary structures are called motifs, and these usually contain about 10 to 40 residues. Up here, a helix-turn-helix motif is shown. These motifs regularly interact with DNA and are found in transcriptional regulators. Patterns found in tertiary structures are called domains. These are independent units of structure that are independently stable. These usually contain about 50 to 200 residues. Here, an alpha-beta barrel domain is shown. This is a stress response domain with an unknown function. Additionally, there are many more ways to classify protein shapes that are conserved in the structural proteome. Here, it is important to understand that the classification helps structural biologists predict the function of proteins based on their shape similarity to proteins with already known functions. The final folded shape of the polypeptide chain and its association with other chains is referred to as the final protein structure. These structures come in all different shapes and sizes, and it's a variety that truly reflects all the cellular work that these proteins are responsible for. Take a moment and pause this video just to appreciate the diversity of the structural proteome.